So today we are going to talk about hemoglobin dissociation curve. Hemoglobin dissociation curve. Dissociation curve, right? What do we mean by this dissociation curve? That how hemoglobin detaches itself or how hemoglobin attaches itself with oxygen. As in my last class, we were talking about the cooperative binding, right? Cooperative binding, I'm just explaining it again. Cooperative binding means that when there is higher oxygen partial pressure is higher, right? What will happen? That the hemoglobin saturation will start taking place. What do we mean by hemoglobin saturation? And with which component? That is, hemoglobin will start binding itself saturation, right? That will take place. This means that the hemoglobin will start binding with oxygen, right? And it will form a temporary compound that is called as oxyhemoglobin, right? And I told you that there are four molecules of heme groups that are present, or hemoglobin one hemoglobin molecule has four heme groups. So we can say that there are four molecules that of oxygen that can bind to the you can say each hemoglobin molecule. So you can say that one hemoglobin molecule, when binds with four oxygen molecules, it is considered saturated and it will form hemoglobin O8. O8 means that there are four molecules of oxygen that are bind to it, right? So in this oxyhemoglobin form. So now, what do we mean by cooperative binding? I told you that hemoglobin has got four polypeptide chains, right? This is the first polypeptide chain. Let's suppose globular protein. It's a globular protein and it has got four globulins in it, right? And with every globular protein, there is one heme group present. There is one heme group present, right? And with every heme group, oxygen molecule can bind itself. So when one oxygen molecule binds with one heme group, this causes the change in the shape of the hemoglobin so that it can expose the second heme group, and so that it will make it easier for the oxygen molecule to bind to the heme group, right? And then the distortion takes place more, and the third molecule of oxygen binds to the third heme group, and then the fourth molecule. So this is what do we mean by cooperative binding. And this cooperative binding is very important so that the hemoglobin saturation can be achieved. And this hemoglobin saturation, this is one of the heme group hemoglobin molecule, and this hemoglobin molecule that is present in the red blood cell is now saturated with oxygen. Saturated with oxygen. Saturated का मतलब आपको पता है ना? That saturation means that when there is no space or there is something that is being completely occupied, so saturated with oxygen, right? Is that clear? So when one hemoglobin molecule, where this hemoglobin saturation actually takes place, I told you in my last class that this hemoglobin saturation takes place inside the lungs. Why inside the lungs? Because I told you that in lungs alveolar cavity, for example, let's suppose this is the alveolar cavity or the alveolar lining of, you can say, the small air sacs present in the lungs, and you know that they are one celled thick like this, right? And each cell is having, you can say, two membranes that an oxygen molecule has to cross. So we can say that when we inhale in air, right? When we inhale in air, what will happen? That the oxygen partial pressure is higher in the alveolar cavity in comparison to the blood that is present in the surrounding of the alveolar lining in confined into the blood capillary, right? So suppose this is the capillary and inside it there are red blood cells, there are white blood cells, there is plasma that is traveling, right? And you know that this red blood cell has got heme group hemoglobin molecule in them. right? So what will happen that this blood that is coming into the alveolar lining, this is deoxygenated blood. I told you in my last class as well. So this deoxygenated blood will when will become oxygenated when the partial pressure of oxygen is high in the alveolar cavity and it diffuses 1, 2, 3, and you know that the capillary lining is also made by one cell. It also has got one celled lining, right? So it has to cross how many layers? Membranes, 1, 2, 3, and 4, and the fifth one will be the red blood cells membrane. So five membranes altogether it crosses and it binds to the hemoglobin molecule. So we can say that the hemoglobin saturation takes place inside the lungs, right, where the oxygen partial pressure is high. And theoretically, we have proved it that if we keep on increasing the oxygen's partial pressure, right? For example, on x-axis, we are increasing the oxygen's partial pressure. And if we look at the hemoglobin saturation, so you will see that the hemoglobin molecule gets saturated when you increase the oxygen partial pressure. And it keeps on increasing and then it becomes constant at one point. So on y-axis, what I have made, this is the hemoglobin saturation, right? And approximately 98% of the saturation will be achieved at higher partial pressure of oxygen, right? And this higher partial pressure of oxygen is usually present in the lungs, right? Are you all getting me? Anas, what do we mean by cooperative binding? Anas, can you
मैम
tell
यहाँ
me
फिर
that
से
what do we
एक्सप्लेन
mean
करा
by
सकती है?
cooperative binding? Means that when the hemoglobin saturation, or you can say the hemoglobin binds to the oxygen molecule, right? One hemoglobin molecule has got four heme groups, and these four heme groups has to bind to the oxygen molecule, right? And this binding basically takes place one by one. So first, one heme group is occupied by the oxygen molecule that causes the shape change in the hemoglobin molecule that allows the exposure of the second heme group so that the second molecule of oxygen can bind to the heme group. So this second binding will cause the distortion again. Distortion means that there will be a change in the shape of the hemoglobin molecule that will take place, right? And that will allow the third heme group to get exposed, and then the fourth one, so on, so that the hemoglobin saturation could take place. And this distortion, this change in the shape, will cause the oxygen molecules to bind to the heme group, right? Means it is making it easier, it is facilitating the hemoglobin molecules to bind to the oxygen, right? So that's why you can say this kind of binding is called as cooperative binding. I hope that's clear to you, Anas, now. Adil, can you repeat it once more? That what do we mean by cooperative binding?
Then when saturated oxygen binds to hemoglobin.
Sorry, I couldn't hear you.
Saturated hemoglobin binds to the oxygen.
No, when the hemoglobin non-saturated, when the hemoglobin binds to the oxygen and it is causing the shape change of the hemoglobin molecule so that the heme groups can get exposed. You need to explain it
Yes, ma'am.
properly. This one line answer is not enough, right? So you should know how to explain it. Anas, what do we mean by cooperative binding?
Ma'am, when an unsaturated hemoglobin molecule binds with oxygen.
Oxygen, how? Explain how. Command term is explain and how. So what you will say that when in explanation you need to give the reason that if you say that they get saturated, but how? One heme group attaches itself with the oxygen, causes the shape change. That causes the second molecule heme group to bind to the second molecule of oxygen. So this is a kind of facilitated binding. And then we've talked about that the hemoglobin saturation takes place where there is higher partial pressure of oxygen. And ऑक्सीजन का पार्शियल प्रेशर हायर कहाँ होता है? ऑब्वियसली जहां ऑक्सीजन का कंसंट्रेशन ज्यादा होता है. ऑक्सीजन का कंसंट्रेशन ज्यादा कहाँ होगा? लंग्स में होगा, जहां पर हम इनहेल इन करते हैं एयर. जब हम एक्सेल करते हैं एयर तो उसमें क्या कहते हैं, ऑक्सीजन का पार्शियल प्रेशर प्रेशर कम हो जाता है. ठीक है, बिकॉज दैट हैज मोर कार्बन डाइऑक्साइड इन इट. सो हियर द हीमोग्लोबिन सैचुरेशन एक्चुअली, जो हीमोग्लोबिन सैचुरेशन है ना, वो इस पॉइंट पे होती है जहां पर डीऑक्सीनेटेड ब्लड आ रहा होता है. एंड इट इज ऑक्सीजन इज डिफ्यूजिंग फ्रॉम द एल्वेलर कैविटी. दिस इज एन एल्वेलर कैविटी, एल्वेलर कैविटी. और इस कैविटी से जब ऑक्सीजन डिफ्यूज करके रेड ब्लड सेल्स तक पहुंचती है, then the hemoglobin saturation takes place. Is that clear? Is that clear, Adil? Ans? Hello? Can you hear me? Okay, I think I should
मैम आप
move
एक
on
और
now.
दफ़ा एक्सप्लेन करा सकती है?
हीमोग्लोबिन सैचुरेशन को देखो, होता क्या है कि जब आपका लंग्स में ऑक्सीजन का पार्शियल प्रेशर ज्यादा होता है, लुक एट दिस ग्राफ हियर. ठीक है, this is showing you the partial pressure of oxygen on x-axis, right, where you can see that the oxygen concentration is increasing, and with that the hemoglobin saturation is also increasing, right? And then it has become constant because there is no more hemoglobin left with no oxygen. So at higher partial pressure of oxygen, for example, at 10, look at this. The oxygen partial pressure is 10, and at this point, the hemoglobin saturation is almost maximum, that is 95 to 98%. So this actually happens in the lungs, where the hemoglobin binds with oxygen, combines with oxygen molecule, right? And this only happens at the higher partial pressure of oxygen. Whereas if you see the low partial pressure of oxygen, like suppose here, the saturation of hemoglobin is less, and where this situation is present, that where hemoglobin saturation will be less, or where oxygen's partial pressure is less, that is in the tissues, respiring tissues. Why? Because the respiring tissues are taking up oxygen from the hemoglobin, right? And at this point, the hemoglobin is detaching itself from the oxygen, and oxygen is being supplied to the cells. And at this point happens in the lungs. But this curve is showing you this hemoglobin saturation where the oxygen partial pressure has increased. We are not considering here carbon dioxide concentration right now. So what am I saying? I'm repeating it again, that when the oxygen's partial pressure increases, right, the hemoglobin saturation also increases, right? That the hemoglobin start taking up oxygen, right, where the oxygen is available, obviously in high amount or in high concentration. So what will happen, that the hemoglobin will completely be occupied by the uh, uh, oxygen, right? And this happens at higher partial pressure of oxygen. So as you can see here at 10, the hemoglobin saturation is this, and it is further increasing at higher partial pressure of oxygen. And this situation is only in the lungs where there is higher partial pressure of oxygen. Now, at low partial pressure of oxygen, this curve is showing that the hemoglobin saturation is less. So where this situation is actually present in the body, in body the hemoglobin saturation decreases where the hemoglobin uh, gives off oxygen, and this situation only happens in the respiring tissues. What are respiring tissues? That are continuously respiring, and they are continuously using oxygen, and they are continuously making up carbon dioxide. So at this position, or in this location in the body, the hemoglobin saturation is least because it is donating oxygen to the respiring tissues. Is that clear? Is that clear now, both of you? Please let me know.
Yes, ma'am.
Okay, very nice. Shall I move on? Shall I move on? Okay, let's
G
move
ma'am.
on with the topic. And such a curve is called as S-shaped curve or sigmoidal curve, right? Like you can see that the hemoglobin saturation is increasing, and this curve is making an S shape, so that's why it is called as S-shaped curve or the dissociation curve that reflects that the atoms of oxygen are binding to the hemoglobin molecule. And how it is binding, I just told you. Bohr shift we will talk about later on. Okay. So now the question arises that what do we mean by Bohr's shift and what do we mean by this is a hemoglobin dissociation curve like we have just talked about, right? Now we are going to talk about the next topic, and the next topic is Bohr's shift and how this Bohr shift actually occurs, right? Bohr shift means that when the hemoglobin that is occupied with the oxygen, like it is saturated with the oxygen, when it reaches to the tissues or the respiring tissues, what will happen that the respiring tissues has to has to take up oxygen from the hemoglobin that is coming in the blood. So let's suppose this is a blood capillary, right, where the blood is coming, and this blood has got red blood cells in here, right, and this red blood cell has got oxygen bind to it. Now what are those factors? This is a red blood cell, right? Now we need to see that what are the factors that will allow the hemoglobin molecule to detach oxygen from it because it is saturated with oxygen, and it has to give off oxygen to the respiring tissues. Let's suppose this is a respiring tissue, or you can say any cell that needs oxygen. Now we need to see that what are the factors through which oxygen will diffuse into the tissues, right? And how it detaches itself from the hemoglobin, right? And this detachment of the hemoglobin from of oxygen from hemoglobin is called as Bohr's effect. Bohr's effect, right? So this is now the topic of discussion, right? Now let's see how this Bohr's effect takes place. Now, for example, I'm making a red blood cells a little bigger to show you certain changes that take place in the red blood cell, and this is present in the capillary, right? And this is the respiring tissue or respiring cell, wherever it is present, right? So when this respiring tissue, right, could be present here or could be in the surrounding, right? When it will give off carbon dioxide, because you know that all the respiring tissues are continuously making carbon dioxide, right? So when it will make carbon dioxide, this carbon dioxide diffuse into the blood capillary, and you can say that it will also reach to the red blood cells. Inside the red blood cell, there are changes that will happen to give off oxygen to the respiring tissues, right? And one of the change is that carbon dioxide reacts with water inside the red blood cell in the presence of an enzyme, and that enzyme name is carbonic anhydrase. The enzyme name is carbonic, let me write it, carbonic anhydrase, anhydrase enzyme, right? And this carbonic anhydrase causes the carbon dioxide to react with water and form carbonic acid, H2CO3. What is this product? Carbonic acid. So actually, I told you maybe in the previous lessons that the carbon dioxide is actually something that is allowing us to breathe more, right? So all this is happening just to take up more oxygen from the lungs, right? So carbonic acid, when will be formed, right? This carbonic acid will dissociate into carbonates, right? That is HCO3 and hydrogen ions. Are you listening to me? Carbonates are negatively charged ions and hydrogen ions are positively charged ions, right? Now, when this reaction will take place, this carbonate has to come out, right? And this carbonate is negatively charged ion. It can only come when the chloride shift will take place. What do we mean by chloride shift? Let me just explain it first and then I will repeat it again so that you understand it well, right? So I'm repeating it again, when the carbon dioxide is released by the respiring tissues, right? And they are giving off carbon dioxide continuously. This carbon dioxide will, when diffuse out of the cells, respiring tissues, it reaches to the red blood cells. And when it reaches to the red blood cells, right? It causes the a reaction to happen in the body, in the red blood cells, and that reaction takes place under the performance of an enzyme called carbonic anhydrase enzyme. So you can say that this enzyme is actually the enzyme that is causing this reaction to happen in the red blood cells, right? Or reaction hai kya, that the carbon dioxide combines with water and it forms an acid called carbonic anhydrase enzyme acid. Carbonic acid, right? Carbonic anhydrase is the name of the enzyme, right? And when this reaction will happen, carbonic acid will dissociate into carbonates and hydrogen ions, right? Are you all getting me? That carbonates,
Yes.
HCO3 negative, and chloride ions that will be formed. And when they will be formed, carbonates will come out of the red blood cells. And this can only happen when the chloride shift will take place. What do we mean by chloride shift? That there is the replacement of the chloride. And can I raise this side a little so that I can write more here? Okay, I'm just writing it back for you. This is carbonic anhydrase enzyme, right? Carbonic acid, when will dissociate into carbonates, HCO3 negative ions, it will also form hydrogen ions, right? And these carbonates will come out of the red blood cell in replacement with, you can say, in replacement with chloride ions. Why? Why these chloride ions come in and why these carbonates go out? Because this chloride shift will allow the charges, it will maintain the concentration of the charges across the cell membrane. That is inside the red blood cells and outside the red blood cells. So that's why there is an exchange of chloride ions that will happen here. So when the carbonate ions will go out and chloride ions will come in, it will maintain the charges across the membrane, right? So when these carbonates will come out, they will get dissolved in the plasma and they will move in the blood plasma as it is. But what about these hydrogen ions? These hydrogen ions are actually creating the problem. These hydrogen ions are, just give me 1 second.
Ma'am,
जी बेटे.
मैम क्लोराइड आयंस डिफ्यूजन से बाहर निकलते हैं, अंदर जाते हैं.
डिफ्यूज नहीं होंगे ना, क्लोराइड आइंस तो एक्टिवली मूव होंगे. दे आर आइंस. आइंस याद रखें, चाहे कहीं पर भी
Okay.
मूव कर रहे हैं, दे आर मूविंग थ्रू एक्टिव ट्रांसपोर्ट. और इट कुड बी अ काइंड ऑफ को ट्रांसपोर्टर प्रोटीन वर्क, बिकॉज क्लोराइड आइंस अंदर आ रहे हैं और साइमल्टेनियसली जो कार्बोनेट्स हैं दे आर गोइंग आउट, राइट? ये
Okay.
क्लोराइड शिफ्ट इसको बोलते हैं. ओके जी. नाउ दीज हाइड्रोजन आइंस आर द प्रॉब्लम. क्योंकि आपको पता है कि व्हेन द हाइड्रोजन आयन कंसंट्रेशन विल इंक्रीज, तो पीएच क्या होगी? दिस हाइड्रोजन आयन विल डिक्रीज द पीएच. पीएच डिस्टर्बेंस क्रिएट हो जाएगी. और जब पीएच डिक्रीज होगी तो ये एक प्रॉब्लम है, दैट इट विल कॉज द disturbance of the blood pH, and this can cause the enzymes to get denatured, Obviously, the enzymes, this carbonic anhydrase will stop working, the other enzymes will stop working, and they will get denatured, and the person may die. So we can say that this decrease in the pH is a problem. And this hydrogen ion will interestingly cause the separation of oxygen from hemoglobin. Why? Because this hydrogen ion will be accepted by hemoglobin present here, and this hemoglobin has got 8 oxygen molecules at, atoms attached with it. So this hydrogen ion will be accepted by these hemoglobin molecules, and this will kick off oxygen to go into the tissue fluid and then to the you can say respiring tissues, right? So this separation of oxygen from the hemoglobin so that it can go into the tissues is caused by the decrease in the pH, or you can say increase in the hydrogen ion concentration. So this, to neutralize it, hemoglobin will act as a buffer. So this action of accepting hydrogen ions, this hemoglobin when combines with hydrogen ions, right? This is a neutralizing effect, or you can say this is giving us a buffering effect, and this buffering effect is very important for our survival and for the enzymes to work best and for the pH maintenance of the blood, right? And this is also important because it is causing the Bohr's effect. This dissociation of the oxygen, or you can say separation of oxygen from the hemoglobin, this is called as Bohr's effect, right? I hope you all got it. Let's see this topic from the book as well, right? So that you get the idea very clearly that what is happening in the blood and in the tissue fluid and in the blood plasma. And with this diagram, we will also I will also detail you that how the carbon dioxide is traveling in the blood. Now, what is happening here? That the tissue fluid has got the tissues, right? And in its surrounding, the tissue fluid will contain carbon dioxide will be that is released by the respiring tissues, or you can say respiring cells, when they are respiring and they're giving off carbon dioxide. And this carbon dioxide, when come into the red blood cell, it reacts with water in the presence of carbonic anhydrase enzyme that forms carbonic acid. And this carbonic acid, when dissociate into carbonates and hydrogen ions, they will be replaced, or you can say they will be exchanged, right? By the chloride shift. Chloride shift is not mentioned here, but yes, when the carbonates come out, the chloride ions go in, right? And this hydrogen ion that is being released, right, this will be accepted by hemoglobin, and this is one of the buffering effect, right? This is buffering effect that is produced by the hemoglobin. Now, on the other hand, you can also see that how the carbon dioxide, rest of the carbon dioxide is being traveled. So this 85% of the carbon dioxide is traveling in the blood plasma in its dissolved form in the form of carbonates, and 5% of the carbon dioxide is moving in the solution in the solution form in the plasma, and 10% of the hemoglobin, the carbon dioxide binds to the hemoglobin to form carb amino hemoglobin. So this compound name is carb. This is also a temporary compound like oxyhemoglobin. Carb amino hemoglobin. And this carb amino hemoglobin compound will take 10% of the carbon dioxide and will carry it in this form. So we can say that hemoglobin also has got an affinity with carbon dioxide as well, right? So this is how this is 10, this is 85, this is 5. Altogether, this is 100% carbon dioxide that is released by the respiring cells. Is that clear to everyone? This is what do we mean by chloride shift.
Yes,
That
yes.
when the carbonate ions are produced inside the red blood cells as a result of action of carbonic anhydrase on carbon dioxide, they diffuse out of the cell and into the blood plasma. These ions have a negative charge, and to balance their movement, chloride ions move from the blood plasma into the red blood cells, and this is called as chloride shift. If what is the importance of chloride shift? If the chloride shift did not happen, the inside of the red blood cells would develop an overall positive charge. Obviously, to maintain the charges, it is very important that and because of the hydrogen ions as well, that the hydrogen ions are also produced. So that's why what will happen, that the overall positive charge will be there in the on the inner side of the hydrogen, on the inner side of the red blood cells, and that causes the pH change, and that should not happen, right? So hydrogen ions cannot leave the cell because its cell membrane is not permeable to them, right? Red blood cell membrane is not permeable, and for that the influx of chloride ions is very important, and this will prevent the change in the overall charge on the inner side, and it will prevent it from becoming more positive inside, right? So what is the importance of chloride shift? Number one, it will maintain the charges, right? Number two, it will help in the removal of hydrogen ions from inside the red blood cells to outside the plasma, so that there is no excessive positive charge inside the red blood cell. Is that clear? To maintain the pH as well, you can say. So this is the advantage of chloride shift, and this chloride shift is very important to remove carbonates outside in the plasma so that it can be carried by the plasma. So this is chloride shift, and this is Bohr's effect. What do we mean by Bohr's effect? That when the oxygen gives off, so hemoglobin gives off or releases